So good evening, everyone, and thank you so much for joining us tonight to cap off our Emanuel Syndrome Awareness Day with this very special time together. This is the last of our speakers for the series that we've hosted to celebrate 25 years of supporting families. I can't believe it's been that long, but it has. We will have one more social coming up Sunday, December 19th at 2 p.m. Eastern Standard Time, which will be another chance to connect with others and see each other from around the world. This day is incredibly close to my heart. Years ago, when Maya was just a baby, I had the incredible luck to connect with tonight's speaker. When researching the 1122 translocation, of which I am a carrier, I found Dr. Emanuel, and in doing so, I found hope. Hope, not necessarily that things would change for Maya, but that there was hope for understanding and connection. Through our years of involvement with Dr. Emanuel, our group, Chromosome 22 Central, has contributed in ways that I am so proud of. Some of us have had the privilege of meeting her in person. My first time was at the Children's Hospital of Philadelphia, and I think it was the year 1999 or 2000.
2000.
Yeah, probably. Others, myself included, have actually sent her blood samples. Having the unbalanced 1122 translocation bear her name has allowed us to have far more reach and connection than we would have had otherwise. Since we named Emanuel Syndrome, there have been 44, I think, new articles that have come out bearing the name, which has allowed us to gather more information. And I'm currently in the process of updating our parent guide. Oh, there's someone from Sweden. This is amazing. So to introduce Dr. Beverly Emanuel, for those who do not know what she does, she is former chief of the Division of Human Genetics at the Children's Hospital of Philadelphia, and she holds the Charles E.H. Upham Endowed Chair in Pediatric Medicine at the University of Philadelphia and CHOP. So Dr. Emanuel investigates diseases caused by abnormalities of human chromosome 22. These include the most common microdeletion syndrome, 22q11.2 deletion, which is today is also, there's a lot of other people wearing red today for the 22q day. And obviously the one that we care about the most today, the recurrent constitutional translocation 1122. So personally, she has been a person I have looked up to for a long time. She has given me the impetus to carry on this work. She has always supported the efforts of Chromosome 22 Central, and I cannot say enough about her. So thank you, Dr. Emanuel, for sharing with our families tonight, and I'm excited to have you speak to us tonight about your lifetime of work on 1122. And I will go ahead and mute myself and let you take over.
Okay, thank you. And thank you for that very generous introduction. And I want to say hello to all of the families, to the children, to the carriers. I'm honored to speak to you tonight on 11/22 of all days. And I just cannot say enough about your perseverance, your dedication, and your willingness, many of you, to allow me to do the research that I have been able to do over many years, and it's still ongoing. So now I will share my screen, I hope. Oops. It is not working perfectly, but let's just, I'm going to click it again. There we go. I'll start just by saying congratulations on 25 years of C22C, and it's really been remarkable. And I thought I would give you a little bit of history of how I got involved and what has brought me to this time. And what I must say is, cut me off if I say too much, because I tend to talk a lot. So here, so my first lab job was many moons ago in 1963 to 64. I actually worked in the laboratory of this guy, Jonas Salk, who discovered the Salk vaccine. And the work was, well, I was in San Diego where the Salk Institute is. The work was fascinating, interesting. I learned a lot about being in a laboratory, but what I really learned the most about was the fact that as much as I loved being in the lab and working in the lab, I really wanted to research the things that were near and dear to my heart and decide independently what things would go on in the lab. So fast forward several years, I got a PhD and then became a postdoctoral fellow in human genetics. My very second paper was on a family with 2 children with what turns out now to be supernumerary derivative 22 or Emanuel syndrome. We got it wrong when we looked at their chromosomes. And the reason was that the extra chromosome that we saw in the mom and in as an extra copy in the children looked different. And we thought it was rearranged, and we thought that explained why there were 2 siblings with this syndrome in one family, and they weren't twins. They were separated by several years. It took us some time. This, by the way, is Paul Moorhead, who was my advisor for those studies, a remarkable man who was one of the people who figured out how to actually culture blood and look at chromosomes. And so I was fortunate to be in his lab, learning how to do chromosomes. And I eventually began to direct the clinical diagnostic lab at the Children's Hospital. Oops. Right. By 1980, we actually got it right. Elaine Zakai and I, Elaine being the clinical geneticist at Children's Hospital, still the clinical geneticist in chief there, we figured out that there was a recurrent reciprocal translocation, the 1122, in unrelated families. And during meiosis, which is the process where egg or sperm are made, one of the chromosomes segregated as an extra copy. And this was published in 1980. This is part of our group. This is Elaine Zakai and myself and Donna McDonald McGinn, as well as several other members of our division. This was at a birthday celebration for Dr. Zakai. Then in 1982, chromosome 22 entered my life again in a different situation, which was the George syndrome. And we figured out with chromosome differences in several children with the George syndrome that chromosome 22 was deleted. These were unbalanced translocations in these 3 children. And this was a story, again, with Elaine Zakai in 1982. So it looked very serious to me that I was leaning toward chromosome 22 very heavily, because running the diagnostic lab, we realized that there seemed to be many more differences of chromosome 22 than many other chromosomes in the genome. And so by 1990, with Jean-Claude Kaplan, I was assigned by HUGO, which was the Human Genome Organization, to actually be responsible for being the gatekeeper of the map of human chromosome 22. And with Jean-Claude in 1990, we had the first international workshop on mapping human chromosome 22. Many people attended, and it was held in Paris. And this is myself many moons ago with Jean-Claude Kaplan at the Maison de la Recherche in Paris. And we had a very successful meeting, kind of positioning genes on the chromosome. And as you all know, genes are packaged in chromosomes, and chromosomes, by that time, we were able to distinguish one from the other. We knew 46XY karyotypes were in the male and 46XX in the female. And those little stripes that you see on the chromosomes are what we call chromosome bands. And that was the fact that banding had been improved was what allowed us to finally recognize the 1122 translocation. And so finally, as part of the Human Genome Project, chromosome 22 became the first human chromosome sequence. So this was a big to-do. It was a publication in our scientific journal, Nature. And if you look at the cover, you see these two fingers about to touch. This was the first chromosome sequenced. We thought it was sequenced from end to end. We know now that there were quite a few gaps, and that's part of what I am trying to close right now. But this led the way to gene discovery. We knew that human chromosome 22 had 49 million base pairs. We got a glimpse of the genes that might be the cause of some of the disorders that we studied. And it allowed us to predict how many genes we needed to study. Little did we know at that time, it wasn't going to be that easy. And actually those fingers touching are from Michelangelo's ceiling of the Sistine Chapel, a magnificent painting, if anyone has had the luxury of seeing it, although we have all seen reproductions. This is, in theory, God creating Adam. And that's how serious and how seriously the Human Genome Project thought the mapping of chromosome 22 and sequencing of 22 was going to be. The interesting part is people ask me, why did you pick 22? Well, part was because of the several disorders that we were involved in studying. And another part was, it's one of the smaller chromosomes. And so we thought it would be easy to deal with. Not so, but that's another story. Now we knew that there were 49 million base pairs, and this was a snapshot of the chromosome with multiple diseases that had been placed on chromosome 22, including some that are now chromosomal in order: DeGeorge syndrome, velocardiofacial syndrome, cat-eye syndrome. Interestingly enough, they did not include any of the unbalanced translocation findings, something that has obviously been of great interest to all of us. However, this is around the time I met Stephanie, and it was pretty exciting because she's put together, which was the first publication directed toward the 1122 translocation. And it was pretty exciting to now have a support group and a publication to refer to. And if you look carefully, this was 1996. So that was the beginning of C22C. And the second one in 1997, yours truly put in an article. And I've kept these near and dear to my heart in my files at work. So thank you, Stephanie, for doing this. It was awesome. Around that time when we figured out the translocation, one of my very close colleagues, also a woman who studied chromosomes, came running into my lab. Her name is Gloria. You can see her at the very top here. And she said to me, oh my God, I think I have leukemia because I can't band my chromosomes. I don't know how, but one of them looks small and unusual. And I said, just give me some of your preparation and I'll take a look. And it turned out that Gloria actually was an 1122 translocation carrier, as was her brother. And one of her sons, who in fact had a daughter who was also a carrier, and her brother has a daughter who was a carrier. And silently, obviously, one of their parents had to have the translocation because it segregated to both of them. So this was another wake up to me that, okay, this is going to be something I'm definitely going to keep studying. And I realized in about May of 2004, they did an article that featured me. And I cut this out because it in many ways says, why have I done this for so long? And my quote was, I found it interesting and perplexing that this small chromosome seemed to be over-involved in many diseases. Why are some children born with a heart defect and others aren't? Why do some have learning differences or a cleft palate and others are normal? And I said, though the genome sequence has been finished, there is still much to learn. And I said, and this is true today, seeing the families we serve, especially their children with problems, really makes me want to get some answers before I finish my career. And in fact, that is part of what has kept me going for these many, many years. I love these quotes from Francis Collins. Francis is now the head of the National Institutes of Health. He was very instrumental in the Human Genome Project. And he said, what is DNA? It's a history book, a narrative of the journey of our species through time. It's a shop manual with an incredibly detailed blueprint for building every human cell. And it's a transformative textbook of medicine with insights, and I just say with insights we hope, that will give healthcare providers immense new powers to treat, prevent, and cure disease. And I know Francis well, and there he is with me. I don't know exactly what year that was, but he certainly has risen to great heights and is directing a lot of the science that is done in the United States these days. And we know that the DNA of the human genome contains many different forms of variants. There are single nucleotide variants, as is shown in this first line, which we call SNPs. There are insertion deletion variants, or indels. There are substitutions of whole blocks of letters. There are inversions, and there are copy number variants. And copy number variants, or CNVs, or low copy repeats, are part of what make chromosome 22 so difficult, one, to study, and so easily rearranged, because it is just loaded with repeated sequence. And we know that SNPs are common in the human genome sequence. There are 1.4 million SNPs in the genome, and those are used to find disease genes. and that we know that the genomes of healthy people are around 99% identical, but major differences are in the SNPs. And we find one every 3 to 1,000 bases, both in the regions that code for genes and in genes themselves. In addition, I mentioned copy number variants and low copy repeats, and there are thousands to millions of DNA bases which vary. CNVs encompass genes leading to dosage imbalances. Sometimes genes are missing altogether because of CNVs, and in fact, that's what happens in the 22q deletion syndrome. And some DNA sequences are repeated multiple places throughout the genome and called low copy repeats. And I will tell you, chromosome 22 is loaded with low copy repeats, which has made it almost impossible to identify the full DNA sequence. And CNVs have been associated with a number of diseases. And what you're looking at up top with every single line, those lines tell us where there are CNVs in the genome responsible for disease. So we are very complicated as a species, and our DNA is just loaded with differences. However, 22q or 22q11.2 has a personality all its own. What you see on this particular diagram are CNVs in the region that we talk about in Q11. In green are the ones that are responsible for the cat-eye syndrome breakpoints, for the 1122 translocation breakpoint, for what we now call 22q11.2 deletion syndrome, or DeGeorge syndrome, velocardiofacial syndrome. And that is the most frequent microdeletion syndrome. And we study that at Children's Hospital. We know from everyone here on 1122 Awareness Day, it is the most common recurrent balanced translocation, which takes place right here, if you can see the pointer. The cat-eye syndrome marker chromosomes have the low copy repeats. And we have found out in my lab that there are numerous other translocations, some which actually are recurrent, that involve this region of 22q. It is really a hotspot for translocations. Many moons ago, we looked at those 4 green blocks of repeat. And this is a paper that was published by Dr. Sheikh in 2000. And I don't expect anyone to read the little text. What you need to see is there are blocks on 22q that have regions of sequence that are almost identical, and they're color coded here. So all of the blue ones are the same, all of the green ones are the same. And if you look right here, there's a huge gap in low copy repeat B, and that's where the 1122 translocation takes place, mainly because it's a region that is extremely, extremely difficult, almost impossible to generate the sequence of. And, let's see. All right, so we know about Emanuel syndrome, or the supernumerary derivative 22. We know that the karyotype has an extra chromosome, and that extra chromosome is the derivative chromosome 22. And there are many, many clinical features that I don't have to really explain to this group because I think you are quite familiar. Although your own child may or may not have all of the features, that's what a syndrome is about. They probably have a good collection of them. These are the chromosomes and the carriers, of which there are many, many, have an 11, which has a little piece of 22 on the end, and this derivative 22, which has a little piece of 11 on the end. And the children with Emanuel syndrome have 47 chromosomes, where that little derivative chromosome is extra or supernumerary. It took us some time to figure it out, but we figured it out. Okay, my mouse isn't working. And these are pictures that I know have been on the website for gene reviews, I believe. And at the bottom is Maya on the left as a baby, and Maya older, and now she's quite a bit older, and I didn't have a more recent picture to put in here. But these beautiful children are the children in your families with Emanuel syndrome. And now we understand a lot more about what causes that translocation. There's a very unusual DNA structure at the junction where 11 and 22 switch pieces, where the two chromosomes join. And that unusual structure leads to unstable DNA when an egg or a sperm are being made. And it's not, and it's interesting because I know many times when a parent has a child with a syndrome, they'll say, is it something I did? Is it something I ate? What made that happen? And the answer is, it's nothing that anybody did. It's our DNA misbehaving. And it would be nice if we could figure out a way to stop that from happening. But we know that in normal individuals, if you, normal males, if you study their sperm, this translocation is there. These are males who don't have the translocation, who are not carriers. So sperm just automatically make this rearrangement. And we've learned a little bit about why that I will come to in a moment. And we also know that the DNA sequence differences between individuals seems to determine the likelihood of a translocation taking place. So there are variations that sometimes make it happen. And in fact, we now have put some of the puzzle together, and I love this cover of a journal from Genome Research in 2007, where on the cover they featured chromosome 22 on the left. In the middle are these unusual DNA structures that we know take place on chromosome 22 and chromosome 11 because of these AT-rich repeats, which I'll talk about in a moment. And on the right are the blocks of repeated sequence, of which low copy repeat B is one of those blocks of just scattered repeated sequence. How it got there, why it got there, we have no idea. And we know that there's extraordinary variation between individuals and between different groups, racial groups, ethnic groups. And we're trying to look at that at this point in time. This is, again, Dr. Zakai, Dr. Sheikh, Dr. Saida, and myself at American Society of Human Genetics meeting, part of the team that kind of put some of this together. And here is Dr. Kurahashi, who with Dr. Sheikh and Dr. Saida, with me squished in between, were responsible for figuring out a lot of the structure of 11 and 22 in that region. And when we actually had meetings in person at the American Society of Human Genetics, we always enjoyed getting together to see what was new on our favorite chromosomes. And so in the year 2000, Dr. Sheikh actually figured out those low copy repeats and the how they are organized, and looked at deletion endpoints in the 22 deletion syndrome. And Dr. Kurahashi figured out that the regions, there are regions that are unstable on 22q and 11q that are really part of the etiology of why this translocation takes place. And basically what he found was that these long AT-rich palindromes were responsible for the 1122 breakpoint. And I'll show you that in a moment. And then I was really curious and worked with Terry Ashley, who is an expert on meiosis, to figure out that, in fact, if you looked carefully at chromosome 11 and chromosome 22 during meiosis, it looks like they sit closer to each other than they do to other chromosomes in the genome. Whether that's accurate or not, we don't know, but it may be that the spatial proximity permits the rearrangement to take place. So basically, what do we mean about palindromic AT-rich repeats? So there are inverted sequences that read the same in either direction. And because of that, the DNA can unpair and push out these hairpin type structures. And the tips of the hairpin are unstable, and in fact, they are sensitive to double strand breaks or breakage, which makes those regions, there's one on 22 at the breakpoint and one on 11q at the breakpoint, makes the region sensitive to breakage and rejoining. Oops. And what's a palindrome? A palindrome is like no melon, no lemon. If you read it forward or backward, it reads the same. And so that's what is going on here, where this palindrome extrudes and then pairs with itself. And then over here at the tip of it, it becomes very sensitive to breakage. And we were curious, and you'll see we've continued to work on that. When we looked during meiosis, and chromosome 11 is painted red, and chromosome 22 is painted green, and where the contrasting dots are the sites of the breakpoints. We did tons and tons of measurements between 22 and 11, 22 and 6, which never, to our knowledge, creates a translocation. And what we found was that 22 and 11 tend to sit closer. So you push that hairpin structure out, you get a break, and they just become friends and join up with one another. And so most recently, Sarah Coral Tash worked in the lab with me as a postdoc. And what she was able to show using specific proteins and enzymes that bind to double strand breaks, that in fact 11 and 22 bind these proteins both during meiosis, when egg and sperm are being made, and in mitosis, when cells are just dividing, they bind more frequently. So the prediction that Dr. Kurahashi made about double strand breakage is actually a real finding. We know now from more detailed molecular analysis. And what's been quite interesting to us, a new 1122 translocation, to our knowledge, we've only seen in males. However, we were contacted recently by an individual who was having pregnancy loss. And when they did chromosomes on her, they found out she has a mixture of cells that are 46XX and cells that are 46XX with the 1122 translocation. And from all the studies that we did, we figured out that this happened after the fertilization that made her, because she has normal and translocated cells throughout her body. So the double strand break sensitivity of these two regions, although extremely rare, can occur after fertilization. And we now have an example that shows it. And there are in the literature 2 other cases like this, but they did not do all the detailed studies that we did to prove that that's how it happened. Most recently, we have been engaged in optical mapping, which is a new technology developed by BioNano company with a specific enzyme that labels DNA at specific sites where there's a particular DNA sequence, and it fluorescently labels long DNA molecules. And then the molecules are drawn into these tiny nanochannels and held in a position. If you think about a spaghetti noodle that you just drop from a height onto, say, a tabletop, it's all curled up. But if you stretch it out, you can make it linear again, and you could mark it at lots of places along its length. And that's exactly what is happening here. And then these molecules are imaged using this BioNano genomics instrument. And basically what it gives us is a barcode, almost a barcoded picture of the DNA molecule. And what we've learned is that this region of human chromosome 22 varies really, really dramatically from individual to individual. I'm going to show you a picture from one of our recent papers. Don't freak out. Oh, sorry. We know that they are very, very large. And these are 2 papers recently with our colleagues in the Netherlands and in Belgium, the top one, and then one coming from my lab where we showed, if you think about this original picture, which I showed you a while ago, it was close, but not quite right, because this is a picture of what, and I don't expect anyone to kind of grasp this completely, 88 genomes, where the red arrows are repeats of some of those repeated structures, and out of 88 genomes, we found 61 different configurations. And this is just one of the low copy repeats on chromosome 22. So it is beyond, it's no surprise that the Human Genome Project couldn't sequence this region, because if you think about it, when you have things that are identical and you break them up into small pieces, they pile up and you only see it as one thing because they're all on top of each other. What this particular technology allows us to do is look at the entirety of the molecule. And this is the work that we are continuing in my lab to try to decipher this complicated region of the genome. And this, by the way, is the team that we're working with now. Wen Tran over here, Andrea Jin, myself, Stephen Pastor, and Mike Z are doing all of this very complicated lab work to decipher what is going on chromosome 22. And hopefully we will get to other regions of the genome as well as we move forward. I could talk a tiny bit about the 22q deletion. As we all know, it's fairly common. There are, and these low copy repeats are really what are responsible for the 22 deletion. It's estimated to be 1 in 3,000 to 4,000 live births. And the youngsters have many, many differences. We thought way back in time that we could detect what was going on by cytogenetics or chromosome analysis. It's impossible to do. There are now better technologies to do that. And these are some of the children with the 22q deletion. A boy in the top row on the left and a little girl in the right, and 2 young teenagers who, believe it or not, look more like each other, even though they're not related, than they look like their own siblings. And that's because we can recognize slight facial differences in these kids. And so what are we doing? 22q is very complex in its organization. And not only is it complex, it is highly, highly variable in structure. We've learned a lot, and we're still learning. We know that these low copy repeats exhibit genomic instability, and they create susceptibility for several types of chromosomal rearrangements, both on 22q and other regions of the genome. And right now we are working with sort of the laboratories of Ming Zhao and Pui Yanquak, who actually designed the BioNano system to take a look at multiple complex regions of the genome. It's unclear whether low copy repeat structure changes by recombination, or that it adds or deletes parts. I think that'll be part of our future studies, looking at large pedigrees with segregation through multiple generations. And we don't know really the role that they play in these different anomalies or abnormalities. The deletion or cat-eye syndrome is something that we will look at, as well as obviously the 1122, which we've been doing. And we've been thinking of this exploration of 22q as a prototype for other complex regions of the genome. And I imagine that 11q will come into play with that as well. And so I'd love to acknowledge really many, many people who have contributed to this work. I kind of showed you the Emanuel lab team, and there are some former members of the team. Our clinical team, Donna McDonald-McGinn and Elaine Zakai for sure, and Blaine and Alice and Daniel. We've been very fortunate to have funding from a number of agencies from the NIH, but in bold here on the bottom are the people that we have to really thank the most, because you've kept us engaged in trying to find answers. The families with the 1122 translocation and the 22q deletion syndrome families. I cannot say enough about how grateful I am to work with all of you to answer these questions, and for your basically resilience and dedication and perseverance. So thank you so much.